think the latest strengths that we're seeing is that in terms of their ability to be predictive and more relevant of the physiology of the human, that is clearly coming on both in academic, but more interesting from our point of view in industry as well. Those technologies are starting to be commercialized, are starting to be used in anger, as it were, within drug discovery pipelines. There are kind of well-known examples where they're already kind of embedded as the standard technology. The important thing to recognize is we're talking here not about cell lines. We're typically talking about stem cell technology, for example. We're talking about people using 3D structures with more complexity in them than just single cell lines in a kind of a monolayer. I think those technologies are now being used actively within big pharma, but also within CROs and also within biotechs as well. So I think what we're seeing at the moment is the advances in the ability to not only build these complex structures in a way that represents, the physiology of the organ or tissue, but also most importantly is this interrogation, as I call it. So the ability to look inside these structures using the omics technology, again, that combined with the ability to build them, I think is really where the industry is going. You can now, and then of course you do that on the AI bit, you've got large amounts of data coming out from complex structures, which you can interpret and look at patterns using AI. You've now got a really, really powerful capability, which probably 10 years ago you didn't have. What we are doing as Newcells in terms of driving innovation in vitro models is we're focusing on what we believe is the biology. We think the real key thing when you're building these complex models is making sure that the biology that you put in is as closely representative of human physiology as you can make it. So we tend to not use cell lines. We focus either on stem cell technology, which is one of our core capabilities, but also we use primary. So we're quite pragmatic in terms of the end goal is to make an in vitro model, which is the most representative and gives you the most predictive data. That's the key goal here. So we're not focusing on one individual technology. We combine them in the ways that we think will give you the best model. And that's how we're driving this together forward. So we're using stem cell technology. We're using CRISPR editing if that's required. We're using bioprinting. We're using flow technology if that's also required as well to build, as they say, the most representative model. The way that these new in vitro models, the complex in vitro models, are impacting on our clients' kind of workflows and their drug discovery pipelines is in a number of ways. I mean, there's 3 or 4 areas that are kind of where we most commonly work are either in efficacy. So this is building a model of disease and then using using their drugs to see whether or not they can in some way affect that progression of that disease, or in safety, but also in pharmacokinetics as well. So understanding how a drug moves in and out of the tissue. It's in all of those areas and actually where these models are actually having an impact. And I think most importantly, it's usually late in discovery currently. So it's usually before they go into animal studies where they're trying to get a rich amount of data about which is the candidate they finally want to take into human trials. So that's where we're seeing these models having a real impact on the drug discovery processes of our clients. In order for us to stay at the forefront of this technology, Newcells continues to invest both internally in developing advances associated with our core models. So we're focusing on retina and kidney and lung. So we can see through interactions with our clients that there are additions or improvements to those models in a number of areas. It could be through higher throughput. It could be through the kind of functional readouts that they're looking at. But they can also be in different types of models. For example, in the retina, we've patented a technology to include an immune component into the retina, which again is something that's been driven from our customers who are interested in the immunology associated with that particular organ. So we do this internally, but we also do reach out to the academic community. We have a number of collaborations with, for example, the University of Oxford working with us on retina, and we have other collaborations looking at our lung model as well, which will help to keep us at the forefront. We work with, I suppose, the technology innovators within the university, as well as feeding in from what our customers are telling us, and then we look at that and decide which areas we're going to invest into to advance our models. I think we look in terms of the broader aspect, strategic, I suppose, landscape of in vitro models, where we see that going over the next 5 to 10 years. I think our perspective is that, I mean, as we said earlier, I think, greater use of those models in all areas, I think, of drug discovery will become the norm. I think we'll see increasingly number of drugs being taken into clinic based on most of the data which is used to get them through regulators will be coming from in vitro models. I think from a technical point of view, I think there is a large focus on linking models together, because obviously, in the human body, all of your organs are connected to each other in some form or fashion through fluid flow. So your liver is connected to your gut is connected to your kidneys, etc. So I think we already internally are looking at models where, for example, we're looking to connect a liver model with a lung model. And I can see this increasingly focus, because that fundamentally is getting very close to the holy grail, I suppose, of an organ, a human body kind of on a chip, where you've got all the major organs of the human body linked and functioning and interacting with each other, which gets very close to, this kind of holy grail, as I call it. I think in terms of our view, that whether or not in vitro models will completely replace animal models, I think my perspective on this, or our perspective on this, is that we certainly can see that happening sometime in the next couple of decades. I think a combination of in vitro models, a combination with AI technology, a combination with improved analytical capabilities as well, and data processing, you can see if all of those technologies converge, you can get to a point where we can mimic the human body in an in vitro environment. And that, I think, means that the requirement for animal studies will be certainly reduced, if not eliminated completely.